How could lifeless molecules have started behaving like something alive? The answer might be simpler than it sounds. They needed a place to stay together. Without any boundaries, early molecules floating in the ocean would just drift apart before anything interesting could happen. But when they got trapped inside small pockets, like bubbles, droplets, or tiny rock pores, they suddenly had the chance to meet and react again and again. Why does that matter? Because every time molecules collide, they can form something new, maybe something more stable or more complex. Scientists today recreate these ancient micro-worlds using microfluidic droplets, tiny water droplets, in oil that act like miniature laboratories. Each one is a self-contained experiment where molecules mix, react, and sometimes even replicate. For example, an RNA reaction that doesn't work in a large beaker starts producing results when it happens inside thousands of tiny droplets, simply because the molecules are no longer too far apart to find each other. A boetius But being in a small space does more than just bring molecules together, it changes how chemistry behaves. In droplets, reactions speed up, random events become meaningful, and each droplet turns into its own world with a slightly different outcome. Some droplets might randomly contain a helpful catalyst or a better mix of ingredients, making them more active than others. Is that not a bit like natural selection in biology? Exactly. The lucky droplets survive and multiply when they grow, fuse, or divide, passing along their chemistry. Modern scientists have even made simple, fatty acid bubbles that grow when fed with more lipids and divide when shaken, all while keeping RNA strands safe inside. It's the earliest form of a life cycle. No DNA, no enzymes, just physics and chemistry doing the job. Now imagine adding an energy source, sunlight, heat, or chemical gradients, and these tiny bubbles start to power themselves. They can capture energy, hold on to information, and respond to their surroundings. That's when chemistry begins to blur into biology. For instance, vesicles sitting across a pH gradient can store energy the way a battery does, and that energy drives new reactions inside. Step by step, droplets that could grow, store energy, and adapt became the first prototypes of living cells. So, could life have started in something as small and simple as a bubble? Surprisingly, yes. And that's exactly what makes the study of microdroplets and protocells so fascinating.
Explore how microscopic compartments and microfluidic technology help us understand the origin of life on early Earth. Compartmentalization is fundamental to life's origin, enabling simple chemical systems to evolve into complex, self-sustaining reactions. Let's begin by understanding why compartmentalization is essential for the chemistry of life. Without compartments, the chemical reactions needed for life would be too dilute and too slow to sustain themselves. In a dilute prebiotic soup, molecules are too spread out, reactions are rare, and products quickly diffuse away before anything useful can happen. But when we partition the same molecules into millions of tiny compartments, the local concentration skyrockets. Reactions that were impossible in bulk suddenly become feasible. Compartments provide several critical benefits for prebiotic chemistry. First, they localize reactants and trap products, preventing them from escaping into the environment. Second, they dramatically increase effective concentrations, making rare reactions much more likely. Third, the boundary can selectively filter which molecules enter and exit, creating the basis for evolution. Here's a protocell analog, a lipid vesicle with a semi-permeable membrane. Small substrates diffuse in, while larger catalysts like ribozymes stay trapped inside, enabling sustained chemistry. On early Earth, rock pores, foam bubbles, and oil emulsions likely provided these crucial compartments naturally. Modern microfluidics lets us recreate and study these ancient compartments with unprecedented precision. Microfluidic devices use T-junctions to generate thousands of water in oil droplets per second. Each droplet is a tiny reactor, perfectly uniform in size. Droplet size depends on the flow rate ratio and the capillary number, giving us precise control over volume and surface area. These systems offer powerful capabilities for studying prebiotic chemistry. We can generate millions of identical droplets in just seconds, creating a massive parallel library of experiments. On-chip operations let us merge droplets, split them, or inject new reagents at exact time points. Each droplet isolates its chemistry from others, preventing parasitic reactions that would ruin bulk experiments. For example, we can screen 1 million different conditions for RNA polymerization in minutes, varying magnesium concentration, pH, and monomer ratios across the droplet library. Confinement doesn't just concentrate molecules, it fundamentally changes how chemistry works. The Damkohler number compares reaction rate to diffusion rate. When it's near 1, droplets become optimal microreactors. In bulk, reactions are slow and inefficient. But in confined droplets, collision frequency increases and yields skyrocket. Random partitioning of molecules creates variation between droplets, giving selection something to act on. When we partition catalysts randomly, some droplets get lucky and receive multiple copies, while others get few or none. This creates a distribution of activities. Even if most droplets fail, a few exceptional ones with multiple catalysts can succeed and dominate the next generation. Networks that would die in bulk can survive through selection. The best droplets grow, divide, and pass their chemistry to offspring, creating Darwinian evolution at the molecular level. Droplets offer additional control mechanisms not available in bulk solution. Evaporation through the oil phase can concentrate reactants over time, mimicking wet-dry cycles on early Earth. Interfaces create local electric fields and pH gradients, adding new chemical possibilities absent in homogeneous solutions. Molecular crowding at the droplet interface can stabilize transition states and RNA templates, dramatically boosting polymerization yields. Real experiments show the dramatic effects of compartmentalization. RNA ligation networks that are too dilute to survive in bulk solution become persistent and productive when partitioned into millions of droplets. Co-encapsulating clay nanoparticles with RNA reveals mineral-assisted catalysis that's completely invisible in bulk experiments. Let's summarize how compartments transform simple chemistry into evolvable systems. Compartments concentrate reactants, create variation through stochastic partitioning, enable selection through differential growth, and support replication through division. Microfluidic droplets give us a tunable laboratory where we can watch chemical evolution happen in real time, testing theories about life's origin. These tools transform vague ideas about prebiotic soup into concrete, testable, evolvable chemical systems that we can study experimentally. Explore how primitive life forms grow, divide, and evolve through compartmentalization and membrane dynamics. Protocells exhibit lifelike cycles of growth and division, mimicking the fundamental processes of living systems. Primitive life requires cycles of growth, division, and heredity to propagate information. Protocell droplets and vesicles grow by absorbing solutes, taking up lipids, or producing amphiphiles internally. Material flows into small droplets, causing them to grow larger, then divide into daughter cells that continue the cycle. Coalescence and electrofusion can mix distinct chemical compositions to create new trait combinations in protocells. When two protocells with different internal molecules fuse together, they create a hybrid cell with combined properties and new capabilities. Physical forces like shear, osmotic shocks, or interfacial instabilities trigger division, partitioning contents between daughter cells. For example, fatty acid vesicles fed with micelles grow and divide upon gentle agitation while retaining encapsulated RNA templates. Template copying and selective partitioning enable evolution in protocell populations. Template copying or ribozyme activity provides heredity, allowing information to be passed from parent to daughter cells across generations. If catalysts or templates bias how contents are partitioned, lineages with higher fitness can expand in the population. Periodic feeding and stress mimic day-night or tidal cycles, imposing selection pressures that favor robust protocells. The coupling of internal chemistry to boundary mechanics makes protocell life cycles experimentally programmable. Microcapsules provide robust shells that protect fragile chemistry while controlling what enters and exits. Microcapsules consist of a protective polymeric or inorganic shell surrounding a liquid or gel core with catalysts inside. The shells provide mechanical robustness and tunable permeability through pore size, cross-link density, and charge properties. Selective transport allows small nutrients and ions to pass through while retaining large macromolecular catalysts inside. Shell chemistries can incorporate mineral phases that mimic prebiotic rock interfaces and provide catalytic sites for reactions. For example, a silica-coated alginate capsule can shelter ribozymes from harsh divalent ion swings while feeding in monomers. Lipid vesicles self-assemble from simple amphiphiles due to the hydrophobic effect, creating flexible boundaries. Fatty acids and phospholipids spontaneously arrange into bilayer membranes, with hydrophobic tails hidden inside and hydrophilic heads facing water. Amphiphilic molecules self-organize into bilayers that close into vesicles, creating enclosed compartments. Membrane properties like bending modulus, line tension, and phase separation govern shape changes, budding, and division pathways. A vesicle can grow by incorporating new lipids, develop a bud through membrane deformation, then split into two daughter vesicles. Proton and ion permeabilities determine whether vesicles can maintain pH and electrochemical gradients needed for energy transduction. Vesicle-to-vesicle fusion and content mixing enable recombination of molecular functions, creating diversity in populations. Coupling membrane dynamics to internal chemistry provides routes to autonomous protocell behavior. Internal chemistry produces molecules that modify the membrane, which in turn affects internal chemistry, creating feedback loops. For example, vesicles with embedded light-driven pumps can maintain a proton gradient long enough to bias phosphorylation reactions. Fatty acid vesicles fed with activated lipids can elongate and undergo shear division while retaining encapsulated templates. This bidirectional coupling between chemistry and mechanics is essential for protocell autonomy and lifelike behavior. These protocell concepts have parallels in everyday systems we already understand. Like sourdough baking, feeding and splitting the starter preserves its character while allowing adaptation to new conditions. Microcapsules are like greenhouses that filter climate stress while letting in light and essential gases for growth. A soap bubble can grow, pinch apart, and merge with others while enclosing a tiny world, just like lipid vesicles. Protocells demonstrate how compartmentalization enables primitive life cycles and evolution. Growth through material uptake, division through physical forces, and heredity through template copying allow protocells to propagate. Microcapsule shells provide mechanical protection and selective permeability, sheltering fragile chemistry from environmental stress. Lipid bilayer membranes self-assemble and exhibit rich physics that governs growth, shape changes, and fusion events. The feedback between internal chemistry and boundary mechanics creates the autonomous, adaptive behavior characteristic of living systems. Explore how early life integrated information, energy, and boundaries to create self-sustaining systems. A protocell is a primitive, cell-like structure that combines boundaries, metabolism, and information processing to achieve lifelike behavior. Let's explore how protocells link information carriers to energy conversion. Origin of life systems must link information carriers to energy conversion and material flux in an integrated network. Encapsulated ribozymes, short peptides, or mineral particles provide primitive catalysis for ligation, phosphorylation, and redox cycling. Activated monomers and high-energy intermediates act as prebiotic currencies analogous to ATP. Chemistries that regenerate these currencies from gradients or light close the loop toward protometabolism. Boundary-mediated vectorial processes, such as proton leakage or ion exchange, couple internal reactions to external fields. Template-directed polymerization in confinement reduces parasitic hydrolysis and increases copying accuracy. Error threshold constraints can be mitigated by selection across compartments rather than within bulk. For example, a droplet containing a photoredox dye can drive NADH regeneration to support ribozyme activity under illumination. Natural gradients provide the energy sources that protocells can harness. Natural settings provide gradients of pH, ions, temperature, and redox potential that can be harnessed by protocells. Microfluidic devices recreate stable, quantifiable gradients for hours, enabling controlled tests of chemiosmotic hypotheses. Vesicles positioned across proton or sodium gradients can accumulate energy via transient pumps or rectifying transport. Channels, pores, and defects in membranes act as primitive valves with selective permeability and gating. Reaction-diffusion coupling generates spatial patterns that can guide growth, budding, or taxis. For example, a vesicle spanning a microfluidic pH gradient accumulates acetate inside and drives an endergonic ligation. Life requires variation, heritability, and differential success, all engineered in compartment arrays. Microfluidic workflows generate genotype-phenotype linkages by co-encapsulating templates with readouts, then sorting by activity. Serial passaging, growth, perturbation, division, and reseeding implements laboratory evolution cycles. Mutational input arises from copying errors, chemical modifications, or combinatorial assembly driving fitness improvements. Environmental schedules with light, feed, and stress impose selection for robustness and autonomy. The result is a practical platform to evolve protocells that integrate boundary mechanics, metabolism, and information processing. For example, sorting droplets for fastest ribozyme amplification over 10 rounds yields lineages with improved tolerance to low magnesium. Bringing it all together to understand the emergence of living systems. The integration of metabolism, information, and boundary physics makes protocells capable of sustained, adaptive behavior. Boundaries provide the essential confinement and organization needed for chemical processes to become coordinated. Environmental gradients supply the energy to drive endergonic reactions and maintain far from equilibrium states. Selection across many compartments refines and optimizes protocell functions through evolutionary dynamics. These three components, boundaries, energy flow, and information, form a self-reinforcing cycle that defines living systems. The transition from chemistry to biology occurs through the progressive integration of these fundamental processes.